The Henry M. Stratton Medal for Clinical Translational Research is presented to Dr. Katherine High of the Rockefeller University. She is being honored for spearheading the development of a gene therapy for hemophilia B, approved earlier this year by the U.S. Food and Drug Administration and Health Canada. This success comes after more than 2 decades of challenging research, during which she directly contributed to the understanding of DNA defects in hemophilia B, factor 7 and factor 10. Dr. High also performed several first-of-the-kind clinical trials in gene therapy, including the first intramuscular and intravascular administration of an adeno-associated viral vector. These studies resulted in adverse events that were not predicted by the animal studies, the basis for which she unraveled in bedside-to-bench laboratory investigations. Dr. High's interest, sparked by a chemistry set she received, she was gifted at the age of 10 and extended to inherited blindness. She was instrumental in developing a gene therapy to restore vision loss in patients with this condition, the first ever gene therapy for genetic disease to be approved by the FDA in 2017. Congratulations, Dr. High. What a pleasure. What an honor and a pleasure. Come on, please. There at the top.
Thank you, Dr. Narla. I would like to thank the Society and its awards committee for this recognition. I have spent much of my career first in academia and then in biotech, pursuing the promise of gene therapy. As most of the audience knows, clinical efforts in gene therapy initially went through cycles of unreasonable optimism, followed alternating with cycles of unreasonable skepticism, both attitudes of which were only partly justified by the facts on the ground. Despite what was going on around us, our own research group moved steadily ahead on our efforts in hemophilia because we believed that none of the problems that we were seeing, that we were encountering, were ones that couldn't be solved. With the support of the NIH, the Howard Hughes Medical Institute, and the Children's Hospital of Philadelphia, we eventually expanded our efforts beyond hemophilia to other genetic diseases, including, as you heard, the rare form of congenital blindness that led to the first approval by the FDA of a gene therapy for genetic disease. The field of gene therapy and now gene editing have a number of approved products now, including for hemophilia and sickle cell disease, and these numbers will only increase. What they share as a class is that they mostly achieved regulatory approval on studies of only a few dozen patients, attesting to the dramatic impact of supplying a normal copy of a gene into the right target tissue. Nobody develops drugs alone, and certainly nobody develops new classes of therapeutics on their own. I want to thank the mentors who trained me, both previous recipients of the ASH Mentor Award, Ed Benz, in whose lab I trained in hematology at Yale, and Harold Roberts, my first division chief. These people taught me scientific rigor and high ethical standards for research, including for clinical research. I want to thank my extraordinary colleagues at Penn and the Children's Hospital of Philadelphia, my colleagues at Spark Therapeutics and then at Pfizer, and I want to thank the regulatory physicians and scientists who labored to build the regulatory infrastructure that is allowing us now to approve cell and gene therapies. Most of all, I want to thank the patients and their families who had the courage to step off into the great unknown in those first trials of gene therapy. Cell and gene therapies and now gene editing are a revolution in therapeutics that is really only getting underway now, and I'm very excited for the future.